Welcome, everybody. It's the third seminar on the Instruct webinar series, so thank you for turning up. As you know, it's Instruct-ERIC is the European Research Infrastructure Consortium for Structural Biology Research, and it focuses on bringing four things like research visits, training, internships, and R&D funding to all the scientists in the participating countries that are Instruct members. Instruct by now has 15 countries that participate, and it has 10 Instruct centers that are offering access to a total of 23 research sites. There's over 9,000 registered users, so we have a good audience, but please do talk to your colleagues. We need people to be aware of Instruct. Our goal is to bring access to our facilities and our training programs to as many people as possible. So please help us to make people aware of what Instruct is and what Instruct can do. In terms of research visits, Instruct offers access to many different technologies that can be grouped in three categories. One is sample preparation for structural studies. One of the flagship technologies, I have to say, is, for example, the nanobody discovery, but also for more things like protein production and crystallization that are a routine for some labs, but might be challenging or alternatives for others. Instruct is there. We have an excellent and extensive collection for techniques of biomolecular analysis that goes to various types of imaging, mass spectrometry, and molecular biophysics. And of course, 3D structural analysis with the three major techniques, X-rays, NMR, and electron microscopy. All these are accessible in multiple sites. Some very specific things that are very difficult to find in your own lab or research sites can be accessible through a peer review procedure, and you are more than welcome. In fact, you're encouraged to submit multidisciplinary approaches that make use of more than one method to lead you towards your scientific aims and objectives. I would like at this point to make a plugin for INEX Discovery. INEX Discovery is a Horizon 2020 infrastructure program that offers services and some training as well, by the way, to things that also Instruct does. The emphasis of INEX Discovery is for 3D structural analysis and a bit of biophysics, but all the things that were in the upper two-thirds of the slides are still unique to Instruct. However, INEX Discovery offers a lot of expertise, offers a lot of methods, and you are more than welcome to look at our web pages. Just simply search for INEX Discovery and use our services. And I should say actually that Instruct and INEX Discovery are collaborating on this project. There's a common access system and there's quite a bit of coordination of things. You can, for example, try to make your sample at Instruct and then come and measure on INEX and so on. So apart from that, Instruct has a few times a year a call for access, extended access, except of the research visits that go through the peer review. It has an extensive training program that, of course, it has been compromised due to the coronavirus situation, but there are still things going on. As you can always check the website for the training events, and you can see like Instruct Never Stops, for example. This webinar series is to make up for the lack of the many workshops that some of them might or might not go on. What I wanted to say also before is that Instruct also funds internships. There are specific calls for that, unlike the common research visits that they're always open. You can always go and submit your call. For the Instruct internships, you can have more extended research visits. It's aimed towards young researchers mainly, to go to one lab, learn one technique, and go back and establish it in their home country. And apart from the research internships, periodically, Instruct publishes a call about research projects that are funding researchers to obtain specific preliminary data that can help them to go on with their research and apply for other funding as well. So please, if you are not already in Instruct, I guess most of you are, but please make sure you register on Instruct using ARIA. And this way you can stay updated for all the calls and for everything that is going on, like these webinars. And please do tell your colleagues about us. We really, one of the main objectives of Instruct is to extend as much as possible the awareness of people that it can offer structural biology facilities and help you advance your research by bringing to it a structural biology perspective. Last but not least, the webinar series is continuing. The next webinar is at the Instruct Centre Spain, and it's, I think, the first webinar of the series that is focusing on COVID-19 issues. So I'm sure that it will be very well subscribed and very well attended, as we were happy to see for the webinar series this far. So I will go on with the schedule and see you in Madrid. So the first speaker is Bert Janssen from the Instruct Centre Netherlands at Utrecht. Bert is a PI there, and he's going to highlight some of his research findings. Bert.
First of all, let me thank you, Tassos, also Instruct and Instruct NL, for the invitation to present the work here. Good morning, afternoon, evening, maybe night to everyone, depending on where you are. I'll start off with showing this drawing from David Goodsell. And David Goodsell, he's based in the US. He's a structural biologist, but he's also an artist, and he makes drawings, paintings, using current knowledge of structural biology, of cell biology. And here he has drawn two cells, pieces of two cells that are interacting. It's part of our nervous system. Perhaps you recognize it. It's a neuron that you see here in blue. It's part of a neuron, an axon, and the axon is interacting with an oligodendrocyte, and the oligodendrocyte has produced myelin, which are layers of membrane around the axon. And there's an enormous amount of interaction between the cells. And these interactions between cells are important for signaling, for the function of tissues, for the formation of tissues. And it's proteins on the cell surfaces that organize these interactions and this communication. For example, in our nervous system, there's an enormous amount of interactions between cells, of neurons that are interacting, for example, via synapses, or neurons that interact with oligodendrocytes that make the myelin. And this myelin is important to increase the speed of the action potential, but it's also providing support to the neurons and the flow of nutrients. And this interaction between these cells in the myelinated axon, but also in the synapse, is important for basic brain functions such as learning and memory formation. For example, there's a protein expressed by oligodendrocytes that sit in the innermost myelin layer. And that protein is called myelin-associated glycoprotein. And this protein interacts with ligands on the neuron membrane, on the axonal membrane. And this ligand is called a ganglioside. It's a glycolipid. And this interaction between the MAG and the glycolipid, the ganglioside, is important for several things. It's important to maintain the distance between the axon and the innermost myelin layer. It's a very well-maintained distance. Because if you knock out MAG, and this is done over 25 years ago, if you knock out MAG, two things can happen. Either the axonal surface collapses onto the myelin surface, which you see here, or the axonal surface dissociates away from the myelin layer. So MAG is able to keep these cells together, but it's also maintaining a very well-established distance. And Dave Goodsell was able to draw this already. I think this is a drawing from 2009, but he hasn't drawn MAG in here because structure is not known of MAG. So MAG is important for adhesion and to maintain distance between the cells, but it's also important to signal, and it signals into the neuron where it inhibits regeneration of the nervous system, for example, arising from spinal cord injury or from a stroke. And it can also signal into the oligodendrocytes where it, for example, regulates myelin formation. So how does MAG do this? And this was a question that a PhD student in my group, Matti Pronker, set out to answer. And he made the extracellular part of MAG, 5 domains, recombinantly in human embryonic kidney cells. And we use a special cell line that produces more homogeneous, shorter glycans, oligomannoses, that we can cut off. So Matti deglycosylated the sample and used reductive methylation, lysomethylation to get crystals. He was able to grow crystals, but unfortunately they don't diffract very well, 3.8 angstrom resolution. The reason for this is probably that they have a very high solvent content, and I'll show you that in the next slide, what that looks like. It's very high. It's only a handful of structures that have such a high solvent content. He solved the structure, and that shows that it consists of 5 Ig domains which are interacting. It's a fairly extended molecule. So the packing of a crystal with 90% solvent looks like this. And for MAG, I'm showing here 1 molecule of the MAG, and in red, and that's interacting, forming this lattice. But you've got these very huge solvent channels there. So when we were measuring the diffraction of the crystal, we were mostly measuring the solvent. And in fact, actually, although it's 3.8 angstrom resolution, the data looks much better than that. It looks more like, well, the data to parameter ratio is more like a 2.5 angstrom crystal structure. And we can see in the crystal that the MAG is dimerized, and it's dimerized via 2 domains, the 4th and the 5th domain, which are close to the membrane. And so we tested whether MAG also dimerizes in solution, and MAG does, but it's very weak interaction. It's a 380 micromolar affinity for dimerization, which is very weak. And this was tested with sedimentation equilibrium analytical centrifugation. So does MAG also dimerize via this interface in solution? And we tested that by making 2 mutations. One mutation that we predicted would disrupt the dimerization is an isoleucine to a glutamate mutation. And we also made a mutation, and I think this is really cool. It shows the power of the predictive power of structure biology. We looked at the structure and we thought, okay, maybe we can make a mutation that enhances the dimerization, because there's a site near the twofold axis in the dimer, where there's a glycan that sterically hinders its symmetry-related dimer. So we thought if we mutate out this glycosylation site, we may get more dimers, and indeed that happens. But first, the monomer. So Matti made the monomer. We collected SACs, the small angle X-ray scattering data of that SACs, and we constructed an envelope of that, low resolution envelope bead model, which looks very, very much like a single subunit in the dimer. And we collected SACs data for the glycosylation mutant, and that reconstruction looks very much like the dimer. So that seemed to have worked. And also in a size exclusion chromatography, we can see that the monomerizing mutant runs at the same retention volume as the wild-type MAG. So the wild-type MAG in this experiment is a monomer because of the weak interaction. But the dimerizing mutant actually runs as a larger species. It comes off early over the column. So we think it's probably a dimer. So how does MAG interact with its ligand, the ganglioside on the axonal surface? And we get a snippet of information from that from looking at the density in our crystal structure, because we have a bit of density which is not represented by the MAG molecule. And this density is near to where others had predicted the ligand binding site to be in the first domain of MAG. And so probably the MAG molecule had something bound from the expression medium, which stayed attached during the purification and during the crystallization. And so we teamed up with the mass spectrometry group, with Joost Snijder and Albert Heck, to see what kind of molecule that could be. And Joost was able to show that about 5% of the MAG molecules, with native mass spectrometry, 5% of the MAG molecules, so this is a bit blown up, had something bound of 854 Daltons, which could be something like a tetrasaccharide. And so we tried to model something in this density, although the resolution is a bit too low. So we decided to go for better crystals and make the first 3-domain of MAG, which contains the ligand binding site. And that crystallizes much better. So we could soak in a trisaccharide that you can buy off the shelf from Sigma. And that revealed much more detailed, 2.3 angstrom resolution of how the MAG molecule binds to its ligand, the GT1B ganglioside. So we could see all the residues that are important, for example, this arginine here, 118. And so we made several mutants to verify this ligand binding site. And to measure these interactions, we designed a new type of experiment where we could mimic what happens if MAG is on one surface and the ganglioside is on another membrane. And we did that by coupling MAG onto a surface plasma resonance chip with its C-terminus. And this is normally used by MAG to couple to a cell via a transmembrane helix. And we incorporated the ganglioside into vesicles, and we've flown over the vesicles over the sensor surface. And that shows that the wild-type MAG indeed interacts with the ganglioside containing liposomes, but most of the mutants do not. And especially the arginine is completely defunct of binding the ganglioside containing liposomes. So is this ligand binding and the dimerization of MAG now important for its function, for its signaling? And yes, it is. And we've shown this by using an assay, a well-established assay, where one probes the signaling of MAG into neurons. And what we've done is we've coated cover slides with using our MAG or our MAG variants. And then we've grown neurons on top of this, and this was a collaboration with Jeroen Passerkamp in Utrecht. And the neurons, they can grow neurites, but MAG is inhibiting these outgrowths. And so a wild-type MAG, or extracellular part of MAG, is indeed inhibiting this outgrowth. And it does so the same as our dimerizing mutant. And we've also made a covalent dimer via an NFC fusion. And also that covalent dimer has the same inhibitory activity as the wild-type MAG. The monomer mutant, though, so the MAG that can't dimerize, is much less active. And also the first 3 domains in MAG, which is lacking the dimerization motif in the 4th and 5th domain, is not inhibiting the neuron outgrowth. Now, the interesting one is that if we mutate out the ligand binding sites with the arginine 118 mutation, and we dimerize MAG, we actually don't get inhibition of neuron outgrowth, but we get stimulation of neuron outgrowth. And we're not sure why this is. Maybe that there's another receptor involved that is affected by this. But it's an interesting observation nonetheless. So how does MAG now sit in between the 2 cells, between the myelin and the axon? And we can build a very straightforward model from our crystal structure because we know how it interacts with the ganglioside, and we know how MAG sits on the membrane because we were only lacking 2 residues to the transmembrane helix in our structure. And so this gives us a model which would show that the spacing between the cells would be about 10 nanometers. And this matches fairly well with recent negatively stained and fixed electron microscopy of a myelinated axon, where this distance here between the 2 cells seems to be very well maintained, somewhere between 9 and 12 nanometer spacing. And then MAG can also signal, as I told, it can signal into the oligodendrocyte, it can also signal into the neuron. Perhaps it's a dimerization of MAG by having this trans interaction and then the cis interaction that provides a signal into the oligodendrocyte. What exactly happens in the neuron, whether it's clustering of the gangliosides or whether there's another receptor involved. It's not clear yet, but that somehow leads to a signaling to the neuron. So it's cis and trans interactions that regulate adhesion and signaling. And now, several months ago, in the summer, Dave Goodsell updated or made a new drawing of his myelinated axon, showing again the axon and the myelin. And now this time he has drawn in the MAG dimer that fits snugly in this interface, keeping the two cells at a very well determined spacing. So I'm very, very glad that this was picked up. So I'm going to shift gears a bit and move from the myelinated axon now to the synapse. And in the synapse, there are many molecules, many proteins that are important for signaling, that are important for adhesion. And one of the protein families that plays a role in adhesion and signaling is the terneurin protein family. Humans have 4 ternurins, and ternurins, they interact in trans. They also form dimers in cis, so on the same cell. And this function of ternurins has been associated with neurological disorders such as schizophrenia and bipolar disorders. So how do ternurins control synaptic adhesion? And this question, Dimfna Meijer, a postdoc in my group, set out to answer. And she first focused on terneurin 3, one of the terneurin variants. Ternurins are type 2 transmembrane proteins, so they have an intercellular N-terminus, one single transmembrane helix, and then a extracellular C-terminus. And for ternurins, there's not much known about the composition. We know that there were 8 EGFs, and these EGFs are actually they are responsible for covalent homodimerization of ternurins. They form cis dimers on the same cell. And there was a very large stretch of almost 2,000 residues that predicted 130 beta strands. That was pretty much it. And then there's a whole bunch of N-linked ligands. And so Dimfna, she made many constructs because we didn't really have a clue of where we should put the boundaries to get a good protein sample. And in the end, she found one good protein sample, which essentially encompasses this stretch of 180 predicted beta strands. She used several tricks to stabilize the protein, and then she tried to crystallize that, but that didn't work, unfortunately. But fortunately, at that time in Utrecht, we had a new microscope, cryo-electron microscope, the Thales Arctica, and the protein is large enough, 220 kilodalton, this bit at least, that we thought would be amenable to Cryo-EM single particle reconstruction. So we used that, we made good grids, and Dimfna was able to get a reconstruction to 3.5 angstrom resolution by using almost 3,000 micrographs. And this already showed that, okay, yeah, indeed, there are many beta strands. Maybe you can see it in the density, the Cryo-EM density. There's also a beta propeller here on the back. And then there's something like that looks a bit like an IG fold or a jelly roll fold. So many beta strands. So that fitted with the prediction. And at the same time, Elena Seredake at the University of Oxford and together with her PhD student, Verity Jackson, they solved the crystal structure of terneurin 2, of essentially the same bit as that Dimfna solved the Cryo-EM structure of. So we teamed up, and we were able to compare also the two structures, and that showed that terneurins have a very unusual fold for an adhesion protein. So it's many beta strands, as I said. For example, you have this very large shell consisting of tyrosines and aspartates. And in it, there is density. There's a long linker that snakes through the sides of the shell and forms another domain. There's a beta propeller on top, and there are two jelly roll folds. So you could see that this core forms a very intricate fold consisting of 7 domains. However, as I said, terneurins are not monomers, but they are dimers. Terneurins form covalent dimers because of two intermolecular disulfide bonds in the second and the fifth EGF. And so Dimfna made the full extracellular part of the neuron 4, that is a covalent dimer, and she showed that on a gel. So not on a reduced and on non-reduced, as you can see, the non-reduced, it runs much higher. And also on a sec, we could see that it's a protein of about 530 kilodalton. So it's a covalent dimer. And Dimfra was able to make grids again. In this case, we collected Cryo-EM data on the Cryos microscope at the Netherlands Center of Nanoscopy. And when we looked at the classification 2D classes, we actually could see that most of the particles showed a monomer back. And this is a bit surprising because we knew it's a covalent dimer, so there must be dimers. All of the sample is a dimer. And so probably these two subunits are very flexibly connected, so we see only one of them back in the reconstruction. Nonetheless, about 15% of the particles, they showed a dimer. And so we reconstructed the dimer, consisting of 36,000 particles to 3.5 angstrom resolution, and this shows how the two subunits are interacting. And the interfaces are mainly organized by the ABD domain that's interacting with the TTR and with the YD shell. And this interface is very hydrophilic, so it's probably necessary to have the covalent connection between the EGFs to give this compact structure. So how is this compact structure also relevant in solution? Because we saw only 50% of the particles having this compact solution, and it appears actually that if you look in solution, most of the particles are in the compact form. So it's probably the freezing of the particles, the air-water interface, that make it go a bit more flexible. And so Wouter Beugeling, a PhD student in my group, we teamed up with Dimfna to look at the SAX data of the neuron 4 dimer, and he showed that indeed most of the particles are very compact. Actually, it's almost, most of it is almost like a globular particle, showing in the normalized Kratky plot. And so Wouter used rigid body modeling using the Cryo-EM reconstruction, and also made models for the bits that we were missing in the Cryo-EM structure, and was able to use rigid body modeling to explain the SAX data, and getting a fairly reasonable model for the full actor domain of the neuron 4 dimer. And this could indicate that how the neuron sits in the synapse, and perhaps it can span quite a long distance, about 20 nanometers, and that is almost the span of the synaptic cleft, which is actually very well maintained at 24 nanometer spacing. So this is a very non-traditional cell adhesion molecule, at least in terms of structural sense. And many questions still remain. So how does it interact in trans? That we don't know yet. And how does it signal in between the two neurons? These are questions for the future. So I've shown you two examples of the work that we do on the MAC molecule in the myelinated axon and the neurons in the synapse. I won't speak now today about other work we've done on SORCES2 and NGF, for example, that induces neuronal apoptosis, and on sortilin, that is important for maintaining homeostasis in our nervous system. So that's for the next time. And with that, I would like to thank the team in Utrecht, and also the collaborators in Utrecht, Utrecht University and the Utrecht Medical Center, Elena Serdak and Verity Jackson at the University of Oxford, and of course, very important, synchrotrons for the beam time, and also NASAN for their access to the Cryos electron microscope. And you all that I can't see that are now looking at the screen, thank you for your attention.
Thank you, Bert. I have to say, Bert, that I always used to say with colleagues that a fantastic compliment to your work is that when it ends up in a textbook, and you showed the alternative to that to be taken up by a professional illustrator, which I think it's a great compliment, actually. But I will let Rolf Bullens answer live. I think, Rolf, you're unmuted.
Hi. Well, EGF domains combine calcium, Bert. Yeah, and so I was wondering, yeah, is this also for 10M3? Yeah, that's conformations would change with calcium binding.
So in these cases, the EGF repeats are don't have the canonical calcium binding sites. But it's actually for many of the adhesion, for many proteins in the synapse, they've been shown that calcium plays an important role in stabilizing the protein, and that there seems to be a role for calcium here for the neurons as well. But that's still work that we are doing. So it could, I mean, calcium is present in the synapse. That there's free calcium present in the synapse, and it looks like that many of the protein molecules in the synapse are using that for stabilization.
So thank you, Bert. We're a bit out of time. We'll move to the next speaker. That's Markus Weingarth, and we will move from X-rays and EM to NMR and antibiotics from the neural system. So please, Markus.
Thank you very much. Well, thank you very much, Tassos, for the introduction, and thank you and Hans Fink for the introduction. And I also like to thank Instruct very much for setting up this webinar series, and I hope that everybody out there is well and safe in these special times. So today I'm going to talk on antibiotics and antimicrobial resistance, and for the sake of time, I guess we can all agree that antimicrobials and antibiotics are important, and I will start right away with the talk. So my talk will be on a special class of antibiotics. These are so-called lipid-2 binding antibiotics. So what is this? The receptor, the target, lipid-2. Lipid-2 is also known as the Achilles heel of bacteria, and lipid-2 is the precursor molecule of peptidoglycan. As all of you know, peptidoglycan forms a protective hull around bacteria, and this hull is essential for bacteria as they die without it. You see here an illustration of peptidoglycan. Peptidoglycan is assembled from strings of repetitive sugar units in blue that are connected via short pentapeptide bridges in yellow. Peptidoglycan is a constant turnover and is constantly maintained via its precursor molecule lipid-2 that you see on the right. Lipid-2 carries one sugar peptide peptidoglycan subunit that is attached to a unique pyrophosphate group and a complex lipid type. Lipid-2 is assembled here in the cytoplasm, then flipped across the plasma membrane, where the head group is chopped off and integrated into peptidoglycan, and the remainder is cycled back for the next round. And this lipid-2 cycle is essential for bacteria so that any drugs that interferes with it is a potential antibiotic. And this notion was famously validated by vancomycin. Vancomycin used to be an antibiotic of last resort, and it binds here at the lipid-2 pentapeptide. However, all of you know peptides can be mutated. That's why many bacterial strains are resistant to vancomycin these days. We work on antibiotics that have a smarter binding mode. Our antibiotics bind here at the pyrophosphate. Since phosphate groups cannot be mutated, this binding mode is ultra refractory to the development of antimicrobial resistance. And it's also very powerful. For example, a few years ago, tioxibactin was discovered. Tioxibactin is an extraordinarily powerful antibiotic, and it also targets lipid-2. I will show you our work on tioxibactin afterwards. So these lipid-2 binding antibiotics are supposed to come in two different flavors. They can either use lipid-2 as an anchor to form deadly pores across the plasma membrane, and the most famous example is niacin, or these lipid-2 binding antibiotics can simply tightly bind to lipid-2 and thereby blocks the lipid-2 cycle. And this is fully sufficient to kill multi-drug resistant bacteria effectively. However, these two binding modes are very poorly understood as a structural level. There are hardly any structural data available and no data at all in membranes. And this paucity of structural data is a major limitation for the use of these powerful compounds to develop antibiotics. It is our ambition to change this. We want to understand these compounds at the atomic level and directly in the physiologically relevant membrane environment using modern solid-state NMR methods. And I want to give you one illustration why we need to understand membrane-active drugs in membranes. This here is niacin, and niacin is the poster child of the lipid-2 binding drugs. There are around 100 papers on niacin every year. Niacin features 5 so-called thioether rings that adapt A, B, C, D, E, and it's well known that rings A and B target the pyrophosphate of lipid-2. And there's already a beautiful structure available, solved by our collaborator EVM working from Utrecht University. This structure was solved with solution NMR in mice cells. Now, all of you know that mice cells are not ideal membrane mimics. That's why we wanted to revisit this complex. And you see here on the right, the original solution NMR spectrum from the NSMV paper that shows niacin bound to lipid tube in mice cells. So let's see what happens if we measure exactly the same complex with solid state NMR in real membranes. And you see this is the difference, light, day and night. The spectra are completely different. And this must mean that the structure of the complex is also completely different. This is something that we published 2 years ago. And meanwhile, we have progressed a lot and we were able to solve the structure of the complex. And we're very happy about this. But today, I'm not going to speak on niacin, but I'm really excited to show you our latest work on tioxobactin. So tioxobactin, what is this? Tioxobactin was discovered 5 years ago in a really spectacular paper that has been cited around 2,000 times by now. And the discovery of tioxobactin really made a tremendous splash. It was globally heralded as wonder drug, as game changer in the combat against multi-drug resistant bacteria. And tioxobactin has a real perspective. It is pretty far in clinical trials. But what is tioxobactin exactly? Tioxobactin is an 11 residue peptide that features many unnatural amino acids, including 4D amino acids, and a funky residue at position 10 called endurocidin. Furthermore, the C terminus forms a cyclic motif. This is the so-called Dapsy cycle, which is strongly suggested to target the pyrophosphate of lipid tube. So what was known on tioxobactin's mode of action before we started? It was known that somehow the N-terminus is important for activity. If you chop it off, then all the activity is gone. Furthermore, it was shown by NMR spectroscopists from Warwick in mice cells that the N-terminus adopts beta-strand conformation in mice cells in the bound state. And it was also known, very interestingly, that you cannot change the order of the DNL amino acids. If you tamper with the order of DNL amino acids, you lose all the activity. Likewise, if you tamper with the order of polar and apolar amino acids, you lose all the activity. So a lot was known already before we started, but there were no data available in membranes, and that's what we wanted to contribute. Therefore, we teamed up with Ishwar Singh from Liverpool University. Ishwar is an expert in synthetic tioxobactins, and Ishwar discovered that you can actually replace this funky residue endurocidin at position 10 by conventional hydrophobic amino acids. This does not reduce the activity, but it reduces the cost of synthesis by 3 to 4 orders of magnitude. So we use these synthetic tioxobactins for our studies. And in order to make these tioxobactins amenable to NMR, we carbon-30 nitrogen labeled 6 of the 11 residues that covered the entire molecule, including the C-terminal Dapsy cycle and the N-terminus. This here is the first solid-state NMR spectrum of tioxobactin in the presence of lipid tube-doped DUPC membranes. The spectrum is already strongly indicative of complex formation because it looks totally different than the free state. And eventually, using phosphorus solid-state NMR, we can unambiguously confirm that we look at the complex. The upper spectrum shows membranes that are doped with lipid 2 in the absence of tioxobactin. The big signal relates to the bulk lipids and the two smaller signals to the two phosphate groups of the pyrophosphate. Once we add tioxobactin, we observe a massive signal shift for the pyrophosphate, which confirms that we look at the complex. Now, with the complex being established, our first question was, what is the secondary structure of tioxobactin in the bound state? Therefore, we acquired this beautifully resolved 2D carbon-carbon spectrum of tioxobactin, and secondary structure is then deduced via so-called secondary chemical shifts, where we compare experimental and random coil chemical shifts. Positive values mean alpha helix and negative values mean beta strands, and you can readily see that the N-terminus adopts beta strand conformation in the bound state. Next, we wondered about the supramolecular arrangement of tioxobactin in the complex. Therefore, we again acquired beautifully resolved 2D carbon-carbon spectra, and blue with a short magnetization transfer time, and a magenta with a long carbon-carbon magnetization transfer time. With a short transfer time, we only see intra-residual contact within the same residue. With a long transfer time, we observe inter-residual contacts over many residues. You can readily see there are many more contacts in the magenta spectrum, and please focus now on the green boxes that I'm going to superimpose. You can readily see there are indeed many more contacts in the magenta spectrum with a long magnetization transfer time. Please ignore signals with blue and black labels. They are not interesting, and focus on the signals with red labels. These are the interesting long-range contacts over many residues. You see here, for instance, long-range contacts between residues 2 and 6, and even between residues 7 and 2, here, there, and there. And if you consider that in a beta strand, the backbone distance between residues A and I+2 is 6 angstrom, it would mean that the distance between residues 2 and 7 is 15 angstroms. And this is simply impossible for NMR, which has an upper distance threshold of 7 to 8 angstroms. So this means these long-range contacts here cannot be intramolecular. There must be between two tioxibactin molecules, which can only be explained by the formation of antiparallel beta sheets. And we could corroborate these beta sheets using fluorescence spectroscopy. Here we used pyrene-type lipid tube. Once two pyrene molecules come close to each other, they yield a unique axiomer signal at 490 nanometer. And you see here unambiguously the buildup of this axiomer signal at 490 nanometer in a concentration-dependent manner. Together, these data compellingly demonstrate that many tioxibactin molecules come together to bind lipid tube. Now we wondered, how many tioxibactin molecules come together to bind lipid tube? That was something that, in collaboration with Evian Burking, we tried to answer with fluorescence microscopy. Herefore, we used MBD-type lipid tube. You see here the uniform distribution of MBD-type lipid tube on the surface of a giant unilamellar vesicle. And very surprisingly, when we added lipid tube, we observed the formation of massive micron-sized lipid 2 tioxibactin clusters. And this was a really surprising, fresh finding. And we obviously wondered, what is the function of these clusters? And we don't know, but we can speculate. And it's very interesting to look at the timeline of cluster formation. The initial binding of tioxibactin to lipid 2 is fast, only requires seconds. But cluster formation is a slow, subordinate process that happens on the time scale of hours. And killing also requires hours. If this is a coincidence or a functional correlation, that's something that we need to investigate in the future. All right, so now we know that tioxibactin forms these massive clusters. So let's look into these clusters at the atomic level. And we first wanted to directly confirm that this Dempsey cycle here really coordinates a pyrophosphate. It was already strongly suggested in the literature, but there was no direct proof. To get a direct proof, we acquired this 2D proton phosphorus spectrum. This here is proton dimension, which shows the amino proteins of tioxibactin. That's the phosphorus dimension, where we have 3 signals from the bulk lipids and 2 signals from the pyrophosphate. You see that the pyrophosphate, but not the bulk lipids, talk to the amino proteins of tioxibactin. How do we now annotate these signals? Therefore, we need to look at a 2D spectrum of bound tioxibactin, and we need to realize that this proton dimension and that proton dimension are exactly the same. And then we can simply transfer proton assignments from the right to the left, and we see that these signals perfectly match with residues 9 and 10 of the Dempsey cycle. This is direct evidence that the Dempsey cycle coordinates the pyrophosphate. And I want to emphasize that these data were acquired at Cher in Florence, first as an INEX project. It was the previous INEX that Wolf Boulens coordinated, and also later on as an Instruct-ERIC project. And together with my colleague from Florence, Moreno Lelli, who is now here smiling in the flowers, we even got funding from Instruct-ERIC for our antibiotics research. So thank you, INEX and Instruct-ERIC, for all the support. So next to the interaction with the pyrophosphate, it is critical to understand how tioxibactin interacts with the lipid 2 head group, because here in the head group, it's where bacteria introduce many modifications, and it's these modifications that convey antimicrobial resistance, just as for the example of vancomycin. But to investigate the interaction with the lipid 2 head group, we had to carbon-30 label it. This was a really tough nut to crack, and I can't thank my collaborator Evian Burking and my former PhD student Dromedary Silver enough to make this happen. You see here a beautifully resolved 2D carbon-carbon spectrum of carbon-labeled lipid 2. These are the sugars and that's lipid 2, so this really works very well. And I want to quickly illustrate on the example of niacin how we can use this to determine the interface. You have here a superposition of 2D carbon-carbon spectra of the niacin lipid 2 complex. In magenta, only niacin was labeled in the complex, and in green, both niacin and lipid 2 were labeled in the complex. And many of these green signals here, without magenta correspondence, provide us with precious information on the interface that we can use for a structure determination. So let's use this approach to determine the complex structure between lipid 2 and tioxibactin. We have here high quality spectra of the tioxibactin lipid 2 complex, and that provide us with clear information on the interface. Here we have plotted all the interfacial contacts on the primary structure of tioxibactin, and you readily see that the interface is indeed localized at residues 9 and 10 of the Dempsey cycle. You also see that almost all interfacial contacts relate to the Murnex sugar, first sugar that is directly present at the interface. There's only one faint signal for the second sugar, which must be distal from the interface. And very interestingly, we did not observe any signal for the pentapeptide in the spectrum. Therefore, you need to know that this is a so-called dipolar spectrum. In a dipolar spectrum, we can only observe rigid residues. The fact that we do not see the pentapeptide strongly suggests that the pentapeptide is highly mobile in the complex. And we could confirm this with a so-called scalar-based spectrum, where we only observe highly mobile residues, and here we only observe the pentapeptide. This is residue 5 and 4, here we have residue 3, and that's residue 2 of the pentapeptide. So the pentapeptide is highly mobile in the complex and not at all involved in the interface. Let's quickly summarize what we have learned so far. We have learned that the Dempsey cycle coordinates a pyrophosphate. The Murnex sugar is directly present at the interface, and the glutamate sugar is distal from the interface. Very importantly, the pentapeptide is highly mobile and not involved in the interface. That's really very important because it's here where bacteria introduce many modifications. Tioxibactin is immune to these modifications. That's one of the major reasons why tioxibactin evades resistance development. Now, the last thing that we needed to understand to determine the complex structure was the membrane topology. Therefore, we used an old trick, the so-called mobility-edited experiment, where we transfer magnetization for lipid tails and water to tioxibactin, and thereby we can determine at the residue level which residues reside in the water and which residues reside in the membrane. And you see that most residues of tioxibactin talk to both the water and the membrane. That's typical for a compound that sits at the water membrane interface. There are only two exceptions. These are here isoleucine 2, isoleucine 6. These long hydrophobic side chains only talk to the lipids because they are partitioned into the membrane. Thereby, we know the membrane topology and we can determine the structure. To determine the structure of the complex, we used HADOC 2.4, for which we got priceless help from this charming gentleman. I'm sure that many of you know him. This is Alexander Weingarth from Utrecht, the lead developer of HADOC. For the structure determination, we used intramolecular distance restraints on lipid 2, intramolecular distance restraints on tioxibactin, torsional restraints. We used tioxibactin, intramolecular distance restraints, and obviously interfacial distance restraints between tioxibactin and lipid 2. And eventually we used a filtering approach, and we only accepted solutions that had the right topology. So that means solutions in which residues 2, 6, and the lipid 2 tail pointed into the same direction. This worked fairly well, and we obtained an RMSD on the backbone of tioxibactin of less than 2 angstrom. This here is the structure of the complex. In yellow, we have the antiparallel tioxibactin beta sheet. You see that the Dempsey cycle tightly coordinates a pyrophosphate of lipid 2. The Murnex sugar is present at the interface, the glutamate sugar is distal from the interface, and the pentapeptide adopts random orientations. Very importantly, we did not observe any specific interactions with the sugars and tioxibactin. And that's the reason why many residues are accepted, are tolerated at positions 9 and 10. And this means that tioxibactin combines tight specific binding to the pyrophosphate with fuzzy unspecific binding to the lipid 2 head group. And altogether, this explains why tioxibactin is able to target many cognate cell wall precursors that all have the pyrophosphate in common, yet slightly different head groups. And this is another mechanism how tioxibactin maximizes its target spectrum and evades resistance development. But what I like really most about the structure of the complex is that it explains the order of DNL amino acids. We have here a clear-cut distribution, a clear-cut separation of hydrophilic and hydrophobic residues. Hydrophilic ones face the water and hydrophobic ones face the membrane. And this is only possible with this order of DNL amino acids. If you imagine a hypothetical all-L Teixobactam, you would not get a favorable distribution. You had some hydrophobic residues that face the water and some hydrophilic residues that face the lipids. So a favorable distribution is really only possible with this stereochemistry. And I know what you're thinking. You're thinking that this is pretty obvious, and I agree in hindsight, it looks totally obvious, but to best of my knowledge, when we started with this project, nobody had this on the screen. So with this, I'd like to come to an end. What have we learned so far? We have solved a high resolution structure of the Teixobactam lipid 2 complex that tells us a lot on the pharmacophore of Teixobactam. The structure also explains how Teixobactam maximizes its target spectrum and how Teixobactam evades resistance development. We have also seen that Teixobactam forms these massive micron-sized clusters on membrane surfaces, and that the timeline of cluster formation coincides with the timeline of killing. What I could not show today are our studies in native bacterial cell membranes. This is a specialty of my lab, so we can directly study antibiotics in native bacterial cell membranes. And in native bacterial cell membranes, funny things happen. You have here an overlay of the Teixobactam spectrum in the bound state in the UPC, and in red directly in the cell membrane of the Staphylococcus strain. And you see there are quite some chemical shift differences. That's something that we're currently trying to understand. So with this, I'd like to come to the acknowledgements. Rhythm Shukla, that you see here on the right, is the first author of and the lead, the key player of all our Teixobactam studies, and João Moderator Silver here in the middle is a key player for our Niacin studies, and João has left us meanwhile for a postdoc in Boston. I also a big thank you to Evian Brooking, our key collaborator, for anything that relates to lipid 2. Again, I'd like to thank Marino Lelli for help with the phosphorus experiment, Nishwa Singh for the Teixobactam synthesis. I like to acknowledge again INEX, Instruct, and Instruct-ERIC for all the support, and you for your kind attention. I'm happy to take any questions. Thank you very much.
Thank you, Markus, for this very interesting perspective, putting things in their natural environment. It changes the views. That's a very nice example for how to connect our work with function. We have questions, and the first one is from Matti.
So my question was, does the structure of the Teixobactam lipid 2 complex suggest any modifications that you could make to Teixobactam to enhance the activity or maybe also the stability?
Yes, it does, but I'm afraid I cannot share this here. Sorry. collaboration with industry and other people.
Correct, fair enough.
That's an excellent question. Thanks, Matti.
But good to know.
Thanks for the diplomatic answer. And we will move to Meindert Lammers for the last talk. And now this will be much more EM than the first talk. And it will be about DNA mismatch repair. So Meindert, thank you.
So today I'll be talking about some insight that we obtained in the last few years about the mechanism of high fidelity DNA replication, and in particular the proteins that are involved in it. So each time a cell duplicates, it needs to replicate its entire genome. And one of the most important thing, or maybe the most important thing, is to do that with high accuracy. You cannot permit yourself to have too many mutations, because as an organism, you will lose your identity. And if you're a higher developed organism like humans, you're very likely to develop cancer. So making sure that you copy the DNA accurately is the most important thing of DNA replication. And this is done in 3 steps, and it starts at the DNA polymerase itself. So the replicative DNA polymerases in all species, from bacteria to humans, are extremely high fidelity DNA polymerases that make, on average, a mistake in every 1 million bases synthesized. Now they do make mistakes. So in addition to the polymerase, there's an exonuclease, which is able to remove any misincorporated nucleotides during DNA synthesis. And the way this goes is shown here in this little movie. This is based on some Cryo-EM structures that we've done previously of the E. coli replicative polymerase shown in orange, the exonuclease shown in yellow, and what's called a DNA sliding clamp in green, which is known as pCNA in eukaryotes. And what you can see is that the DNA very efficiently moves from the polymerase active site to the exonuclease site in order to remove the misincorporated nucleotide, and then DNA synthesis can resume as normal. So this is very efficient and very fast. It happens in milliseconds time scale. Problem arises when mistakes are not removed in time, because then they get incorporated into the DNA, and that means that it will introduce mutations. And the way to resolve that becomes vastly complicated, because when you look at a mismatch, there's nothing wrong with the nucleotide per se. It's just that it's in the wrong base, or in the wrong place, sorry. So the question becomes, how do you recognize the wrong strand or the wrong nucleotide from the template one? Now, in order to do so, there's a complicated mechanism called DNA mismatch repair, which includes many different proteins that both scan for the mismatch, as well as trying to find a strand discrimination signal, which is able to discriminate between the template strand and the newly synthesized strand. So a lot of research has explained to us how this might work. So in broad strokes, we can explain the system in a few steps. Initially, there's a protein called MUTAS that will scan the DNA many, many million bases in search for a mismatch. When it finds a mismatch, and upon binding of ATP, it will then undergo a conformational change that will allow it to recruit a second protein called MUT-L. Then together, MUT-L and MUTAS will then search for the strand discrimination signal, which in E. coli is a methylation site, or GTC sites that are methylated. And so in a brief time window between DNA synthesis and methylation, at these sites, you will find the template strand being methylated, whereas the newly synthesized strand is not methylated. And this is the signal for a third protein called MUT-H to be activated by the MUTAS-MUT-L complex and incise only the daughter strand. This in turn then becomes the entry site for UVRD helicase, as well as the exonuclease or RECJ, depending on the direction the excision will take place. So together, the large complex will then remove the newly synthesized strand for up to 1 kilobase pair in length until it finds the mismatch again, which it will now remove, and then somehow it's signaled to release itself from the DNA, and then recruit the DNA polymerase again to resynthesize DNA, hopefully this time without a mismatch. So it is clear from the scheme that MUTAS, here shown in green, plays an essential role in the whole repair process. It scans for mismatches, it recognizes, it recruits the partner protein, and it initiates and guides the whole repairs cascade. So how is a single protein able to do all these different tasks? So about 20 years ago, we determined the crystal structure of MUTAS bound to the mismatch, which is a key step in the repair cascade. But ever since, we've not been able to understand all the subsequent steps in much detail. So recently, we've started looking at this again, now using Cryo-EM. And along the way, we determined again the structure of MUTAS bound to a mismatch, like we did 20 years ago by crystallography. And it's always good to see that when you then place the crystal structure into the Cryo-EM map, you find that the two actually superimpose very, very well. And the only difference is that when you turn around the map, you can see that in the Cryo-EM map, we have a bit longer DNA because the substrate used for the crystallography was a lot shorter than that used in the Cryo-EM. The structures also reveal how MUTAS recognize the mismatch, and it does so by kinking the DNA, as you can see here. The DNA is kinked is about 60 degrees, and by doing so, it opens up the minor groove. Let me get to the right one, and thereby exposing the mismatch and allowing it to form a hydrogen bond to one of the mismatched bases and insert a phenylalanine opposite the mismatch pair, and therefore lock the MUTAS onto the DNA at the site of the mismatch. Now, a few years later, about 5 years ago, a new structure was determined by the group of Titia Sigma, where there's a complex of MUT-Ls bound to a fragment of MUT-L, representing the complex after mismatch recognition and ATP activation. Now, normally this only happens on DNA, and while DNA was present in the crystallization trace, it was not visible in the or in the electron density map. So to understand how this complex arises and how it interacts with the DNA, we then use Cryo-EM to determine the same structure, hopefully now revealing the position of the DNA in the complex. And what I'm showing you here is the Cryo-EM map of mutase, mutal, and DNA. It's a bit low in this stage. We have a high resolution map now, but the essential features are conserved between the two. So for now, I'll just use this map. And here you can see the DNA clearly running through the middle of the complex, in which we can easily place the different proteins. In red, or in green and in blue, you find the two mutase monomers wrapped around the DNA, which is going straight through the whole of the complex. And on the left side now, you can see the fragment of mutal called LN40, which interacts with both the DNA and the two mutase monomers at the same time. One thing that's really striking is that although the two mutase monomers are identical in sequence, they adopt different conformations and different positions on the DNA. And only one mutal monomer is interacting with the mutase dimer. This is because the distancing between the two mutase monomers relative to the DNA is slightly off, which implies that one monomer here shown in blue is actually interacting with the minor groove, whereas the other monomer in green is interacting with the major groove of the DNA. And this therefore allows the mutal to bind only on one side where it can interact properly with the DNA, whereas on the other side it would cause a clash with the DNA itself. This is not because we used a short DNA, because the actual length of the DNA is much longer than we could see in the Cryo-EM map. This is simply because the mutase dimer is positioned asymmetrically onto the DNA. We also find that one of the mutase monomers, the one in green, is wrapped itself around the DNA to enhance the DNA interaction surface and get a better grip on the DNA. Now, this structural symmetry was quite surprising, but not entirely new, because in the early structure of mutase bound to a mismatch, we also found that only one of the two monomers interacts directly with the mismatch, whereas the equivalent domain in the other monomer is interacting non-specifically to the backbone of the DNA. So there's a conserved or continued asymmetry within the symmetric mutase dimer, which is interesting because later in evolution, there we find true heterodimers, which consist of two mutase homologs in which the two monomers do really play different roles within the repair cascade. But the true surprise comes when we actually look at the differences between mutase bound to a mismatch and mutase activated and binding to mutal. And that's emphasized when we do a morphing between the two states, where we see a huge conformational change within the mutase dimer, where a large domain is moving by about 180 degrees, rotating away from the center to the bottom of the complex. This is caused by closing of the two mutase monomers, which by closing push down the DNA, which in turn is pushing down the other domains. And by doing so, it creates a binding site for mutal, which previously would not be able to interact with the mutase dimer. But this now raises the question, why doesn't this happen on normal DNA? Why does it only happen on mismatched DNA? And to answer that question, we had to do a trick, because when mutase is scanning normal DNA before it finds a mismatch, it actually translocates at high speeds along the DNA. So this very mobile molecule is hard to trap for structural studies. So what we did here, and I hope you can see it, this is a micrograph, electron micrograph of DNA molecules, long linearized plasmid DNA, with mutase molecules scattered all over the DNA like little beads on a string. And just to emphasize what it looks like, I've drawn in some of the DNA molecules, as well as the mutase molecules, which are scattered all over the DNA. And so from this micrograph and many more, we were then able to reconstruct the Cryo-EM map of mutase while it's scanning for DNA or for a mismatch. And this showed us a structure that looks very much like the mismatch state, but is different in some essential features. So when we again do a morph between the scanning state and the mismatch recognition state, we see a few differences. The first one is that the two monomers close somewhat, they get more compact, and the DNA is being kinked from a straight into the kinked state where it's about 60 degrees bent by the molecule. A smaller conformational change is actually a very essential step, which is where we look at the right side here in the movie. You can see that the blue monomer is moving or translocating relative to the green monomer along the length of the DNA. And this is a crucial step in the activation of the repair cascade. And I'll highlight a bit more in the following slide. So when we take a top view of the mutase dimer, we can see that the two monomers are juxtaposed one another, embracing the DNA. So they're not able to make any closing because the DNA is forming an obstruction to do so. Upon mismatch binding, where the DNA is being kinked, the two monomers can now reposition each other alongside the DNA. And you can see that the blue monomer has moved from the left all the way over to the right. And this is a crucial step because in the next conformational change, these two monomers need to slide alongside one another, like so, thereby pushing the DNA down, causing the domains of the mutase to rotate and create a new binding site for mutal. So it's this small conformational change between scanning of the DNA on homoduplex DNA to the mismatch recognition, which kinks the DNA and allows the repositioning of what we call the clamp domains, that then allows the conformational change that is needed to push the DNA down and create a new binding surface through mutal and thereby initiate the repair cascade. So in summary of those movements, we go from scanning to a mismatch recognition, which sets up the repositioning that allows then the conformational change that drives the repositioning of the connector domain and then creates a new binding surface for mutal that would not be possible during the scanning of the DNA. But this then begs the next question: what's actually driving all these conformational changes in mutase? Is this simply just the DNA, or is there something else going on? Well, we know that there's something else going on because right at the other end of the molecule, on the other end where there's no DNA binding, are two domains that are HPase domains belonging to the ABC superfamily. And these are typical dimer HPases. They're found in membrane transporters where the ATP hydrolysis is used to transport molecules across the membrane, but they're also found in DNA repair and DNA conversion molecules like RAD50 and MRE11, where the HPase domains are connected to a very, very long coiled coil that wraps around the DNA and helps it stay together or condense it during DNA compaction. And the reason why these molecules, these very diverse molecules, make use of these ABC HPases is because the ABC HPases are composite active site, which means that in order to complete the active sites, the two monomers each contribute to a complete active site that only upon closing of the mutase dimer, or ABC dimer, I must say, is able to hydrolyze ATP. So by binding and hydrolyzing ATP and then afterwards releasing of the ADP phosphate, you have a little motor that is moving continuously or under control of other factors. So how does this little motor then affect the conformation of mutase? And for that, we then determine the structures of mutase without DNA, in the presence of ADP and in the presence of ATP. And you can see there's a huge difference in between the two structures. So this is entirely identical molecule, and the only difference is a single phosphate and 4 oxygens in the ADP or ATP molecule. And on the left, you see the ADP map. In orange is the map. It's very low resolution where we can fit in the molecule, but we don't see much detail. And the reason for that is we can see from negative stain 2D classes is that the top domains are extremely flexible. And therefore, trying to get an average structure out of the data results in a fading out of the electron map due to the flexibilities in these top domains. In the ATP state, however, there's a big difference. Now it becomes a very stable molecule where we get a beautiful map where we can see individual side chains as well as the ATP nucleotide very well defined in the Cryo-EM map. So when we take these structures again and do a morph between them, we can see that the native conformational changes that mutase needs to undergo whenever it wants to hydrolyze an ATP molecule. So ATP binding and hydrolysis results in a movement, an opening and closing of the mutase dimer. And it needs to open up in order to bind DNA, because in the closed ATP state, it's not able to wrap itself around the DNA. So when DNA comes, it suddenly becomes a steric block to the ATP closing. It cannot hydrolyze ATP now because it cannot undergo the conformational change it needs in order to create the complete active site for ATP hydrolysis. So the mutase dimer is now locked on the DNA and it cannot release itself from it. So the one way out is to find a mismatch where it can actually kink the DNA, undergo the conformational change that is needed to then go into the next step where it completely closes it, pushes away the DNA and creates a new binding site for mutal and thereby initiates the repair cascade. So mutase is a central protein within the repair cascade that performs many different functions. And these many functions are allowed because it can adopt many different structures. So one single protein exerts many different functions by adopting different structures. As shown here in this scheme, in solution, MUTS transforms between closed ATP bound state and an open ADP bound state. Then when it's bound to DNA while it's scanning for a mismatch, it is a somewhat open disk that is trapped around the DNA. Upon recognition of the mismatch, it undergoes a conformational change by bending the DNA at the site of the mismatch. And that then is the stepping stone for the next conformational change that creates what we call a sliding clamp that recruits mutal and is then able to look for the strand discrimination signal, which is in this case MUT-H bound to a hemimethylated site. Next, the recruitment to UVRD and the exonuclease is something we're working on to find out how the next step is working. But somehow this is then able to translocate back along the DNA in search for the mismatch, removing the newly synthesized strand and also thereby the misincorporated base. After that, the polymerase is recruited and start synthesis again. And for that, we have multiple structures as well. Here, the E. coli polymerase in orange again, and the exonuclease in yellow, and the clamp in green, which will then synthesize the DNA as it's supposed to do correctly this time. So clearly, in order to execute the different functions of the mismatch repair cascade, you need different structures of the same protein, allowing it to adopt different functions and states, and so travel through all the way from the beginning to the very end of the repair cascade. Most of the work I've talked about now was done by Rafael Leiru, who's now a group lead at the CNAO in Madrid. And this was in collaboration with my former PhD supervisor, Titia Sixma, and a new or a recent PhD student, Dorette. And this is actually part of a larger collaboration, an ITN-supported DNA repairment project, which involves many different people performing DNA repair at different levels of biochemistry and structural biology. My lab here in Leiden now is continuing some of the work in mismatch repair as well as other studies. And the last two structures that I presented of the ATP and ADP states were done by Alessandro Borsellini, who is a very, very talented PhD student who's collecting more and more Cryo-EM structures all the time. And this is done, here's my last slide, at Nissen, which is just 5 minutes away from my lab. Where we recently upgraded one of our cryos with a K3 camera, and we're now regularly collecting 300 images an hour. So if you're in need for Cryo-EM time, do call us up, or you can access us also through the Instruct-ERIC website and INEX. So I hope to see you in Leiden someday soon. And with that, I'll take any questions.
Thank you, Meindert. Moving us back inside the cell with quite a few structures and states and so on. Sylvia, you can unmute and you're allowed to talk.
Hi, I was wondering whether all mismatches are recognized with the same efficiency. You know, from your structural model, I would have expected to see differences between possibly different type of mismatches, or all of them cause the same bendability.
So they do show different affinities. So some are better recognized than others. But multiple structures of MUTS bound to different mismatches show that the type of bending and the interaction are conserved. But I think it's the feasibility of the bending that is easier on certain mismatches than it is on others that will that is causing the selectivity for ones over the others. But the final structure will be the same.
There's another question from Jose Maria Carazzo. So when you have continuous flexibility, not everything is lost in Cryo-EM. Instead of averages that cannot work, have you considered computing trajectories? The most simple approach is possibly principal component analysis. See our recent work on COV2 with Tagare or CryoSpark work.
Yeah, so we have actually done principal component analysis on the scanning state because it also shows a degree of flexibility. The challenge here is that the domains that are moving are so small that at some point there's nothing to hold on to. These are the domains that we're looking at are maybe 15 or 20 kilodaltons. So that's simply not enough to really robust do subdomain averaging. But for the larger, if we look at this, if we compare the two MUTS monomers as a whole, then we can do principal component analysis on their relative orientation. Yeah.
Thank you, Meindert. We have one more question that Isabel Martin is allowed now to talk.
Thanks for the talk. I enjoyed it a lot. I observed really quickly that your group also works with tuberculosis. So I was wondering if you heard about the protein NUCs, the non-canonical mismatch protein, and if so, if you have any ideas on how if it may work on a similar fashion to the mutase-mutel complex.
So I do not work on NUCs. I am very much intrigued by it. So for those who don't know, mycobacteria are the only species that we know of that do not have the canonical mismatch repair. And so that was very puzzling. And until recently, somebody found a protein called NUC-S, which also seems to be involved in mismatch recognition and repair. Little is known about it. It tends to bind to the mismatch and maybe even incise next to the mismatch, but the details are not known. And frankly, I have not dared to step into it because there were several other people already working on it. But I haven't heard anything about it in the last few years, so maybe it's time to pick it up again. But my work on TB is not on the mismatch repair in mycobacteria. No.
Thank you, everybody. This thanks to the speakers, of course, to Bert, to Markus, and Meindert. I hope you're all there in the next webinar. You can go to the Instruct site and decide. So thanks again to everyone for attending. Thanks to the speakers, and see you all at the next webinar.